Welcome back, and this is the last in the series of videos looking at inorganic chemical applications. In this video we're going to be looking at two main things. We're going to be looking at how to stabilise an unstable oxidation state. We're also going to be looking at complex ion examples. The two examples we're going to be looking at are cisplatin, a cancer drug, and hemoglobin. So we're on page 12 of your notes. And we're looking at how we would stabilise an unstable oxidation state. In this case, we're looking at chromium 2. Now, chromium 2 is the blue chromium species. It's very easily oxidised to chromium 3, which is the green solution. In fact, all it would take was exposure to air, because chromium 2 can be aerially oxidised to chromium 3. However, if we complex it, in other words, if we turn it into a complex ion, we can kind of trap the chromium in the +2 oxidation state. And that's what we're going to do with this experiment. So we have this set up. Here we are generating our chromium 2+ by reacting sodium dichromate zinc solution with some acid. Once this has gone blue, we use the pressure from the hydrogen, which is also being released, to push the chromium 2 species up and into the sodium ethanoate solution. When it reacts with that sodium ethanoate solution, it complexes and forms a red complex. So from our data book, there are three equations that we are interested in. We are interested in the zinc equation, 2 electrons going to zinc. Remember, the data book is always written as reduction potentials. We're also interested in the dichromate, Cr2O7. 2-, this is one we've come across before, plus 14 H positives, which is why we need that acid present, plus 6 E negative going to 2 Cr, in this case 3+, plus 7 H2O. But we're also interested in the Cr3+ going, so that should be reversible, going to Cr2+. Okay, so those are the three equations we're looking for in our data book. And when we combine the equations, we're using zinc as our reducing agent because it's very easily oxidized. And when we combine the two half equations, we get positive E cell values for both the first and the second reaction. So the zinc should be able to reduce the dichromate from the +6 oxidation state, first of all to the +3, and then from the +3 to the +2. Now, if we take a little of the chromium 2 solution and let it stand in air, that blue chromium 2 turns green via aerial oxidation. But over the same period of time, the chromium 2 ethanoate stays red because the complex ion is stabilising that unstable oxidation state. So on the next page, we're looking at the first of our complex ions. And this is the complex ion we're looking at. It's called cisplatin. So this is a platinum complex with 2 chloro groups, and the 2 chloro groups are on the same side of the molecule, which is why it's the cis version, and that's very important. I've got 2 ammonia groups, again on the same side, and it is only the cis version that has any effect. The trans version is not effective as a cancer drug. So you'll notice that this square planar platinum complex is neutral, so it diffuses through the cell wall, or it's taken through active transport through the copper channels. Once inside the cell, however, this is where the drug gets activated. It's activated by the fact that it loses a chloro ligand and it gains a water ligand. So you now have this, and this is the active principle. This is actually the active drug. It now has a positive charge and it has a water molecule in place of one of the chloros. Now this molecule can now bind to the guanine of a DNA. It pulls the DNA helix out of shape by cross-linkages, and it prevents the DNA from replicating, and hence the cell dies. And that's the important principle of how this works. As we said before, it's a square planar structure because it can produce EZ or cis-trans isomers. It has an oxidation number of +2 and is called the diamine. Notice the double M again because it's got ammonia molecules. Dichloroplatinum 2 because the oxidation number of the platinum is +2. And it's this square planar shape. As we said before, it can diffuse across the cell membrane because it's neutral. It can also be taken through the copper channels. It's a compound which has the actual medical effect. Remember, it doesn't have that effect outside of the cell. It's only when it comes inside of the cell and you replace one of those chloroligands with a water ligand that the medicine actually becomes active. Now that we have a charged molecule, there's a much stronger attraction between those and the polar water molecules, and therefore it stays within the cell. By causing cross-linkages across the DNA molecule, it pulls that molecule out of shape, and therefore the DNA molecule cannot successfully replicate and divide into two cells, and we end up with cell death. The nitrogen and oxygens have lone pairs of electrons, whereas the carbons and hydrogens don't, and so they can't form these cross-linkages. The reason this targets cancer cells better than normal cells is because the cancer cells are replicating much faster. However, it will be taken up by all cells in the body, and this is why it causes side effects for things like hair loss and stomach problems, because there are cells in that part of the body which are replicating quite quickly. The last complex ion we're going to have a look at is hemoglobin. And this is the complex ion with iron, and specifically the iron 2+ at its centre, and it's forming dative covalent bonds with the nitrogens of the heme molecule. Now, there are two states of the heme molecule, one without oxygen and one with oxygen. Obviously, where there are high concentrations of oxygen, for example in the lungs, this equilibrium position would be pushed forwards and the heme would accept the oxygen. Once it moves to a cell where there's a lower concentration of oxygen, the equilibrium position moves to the left-hand side, releasing that oxygen. The problem with things like carbon monoxide is they will bind irreversibly onto the heme molecule, making it very toxic. And this is an example of ligand exchange.